Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity. My name is Ikayanti Marfuatu Solihah. I'm a researcher from Research Center for Pharmaceutical Ingredients and Traditional Medicine, National Research and Innovation Agency, or Pusat Riset Bahan Baku Obat dan Obat Tradisional, Badan Riset dan Inovasi Nasional. Today, I would like to present my research, which is titled Cytotoxic Activity of Akalifa wilkesiana, Ziziphus numularia, and Glossidion zeylanicum on 41 breast cancer cell line. First, I want to start with the background of the research. As we know, cancer is the second leading cause of death worldwide. Based on the WHO data, breast cancer is the #1 cause of death both in developed and developing countries. Previous research found that akalifa or Akalifa wilkesiana leaves, bidara or Ziziphus numularia herbs, and Glossidion or Glossidion zeylanicum leaves, water extract have potential effect on breast cancer cell line. The study was aimed to examine the effect of the ethanol extract of medicinal plants on cell viability, cell cycle, and apoptosis in human breast cancer 41. These are the methods of the research. We collected the plant materials from different areas. We can see the picture of the plants below. The first picture is akalifa, collected from Tawangmangu, and the second picture is bidara, collected from Trisik Beach, Kulon Progo, and the last picture is Glossidion tree, collected from Magelang. The dried material were macerated using 96% ethanol for 72 hours. Cytotoxic assay against 41 cancer cells was evaluated by MTT-assay. Cells in 96 well pad were treated with series of concentration of extract for 24 hours. Fractionation was done by liquid-liquid partition using hexane, ethyl acetate, and methanol. Cell cycle profile and apoptosis induction of the most potent extract was observed using flow cytometry. We used linear regression to analyze the data of cytotoxic assay. The linear regression shows the correlation between concentration and cell's viability to obtain IC50 value. The flow cytometry data was analyzed using BD Accuracy J6 software. Now let's move to the results. Based on cell viability, each extract treatment showed a linear relationship between concentration and inhibitory activity of cell proliferation. Table 1 showed that the ethanol extract of akalifa leaves had the strongest cytotoxic activity with IC50 values below 200 microgram per milliliter, exactly 171.83 microgram per milliliter. Two other test ingredients, ethanol extract of bidara herb and Glossidion leaves, showed IC50 values above 200 microgram per milliliter. We can conclude that akalifa extract is the most potential extract developed as a chemopreventive agent. Furthermore, akalifa extract was fractionated by the liquid-liquid partition method using hexane, ethyl acetate, and methanol multilevel solvents to separate compounds based on their polarity. Each fraction was tested for its cytotoxic activity against 41 breast cancer cells. The IC50 values of each fraction are summarized in Table 2. The results of the cytotoxic test showed that the hexane fraction had the highest proliferation inhibitory activity with an IC50 value of 85.02 microgram per milliliter, the least compared to the other two fractions. This means that the hexane fraction is more potent than the other two fractions. The potential of this fraction needs to be tested for its cytotoxic activity against normal or non-cancerous cells to supplement its safety data so that it can be further developed as anti-cancer agent. Observation of cell morphology shown in Figure 1 shows that akalifa extract resulted in changes in cell morphology compared to cell control. From Figure 1, it appears that there is inhibition of proliferation in cells treated with akalifa extract. Changes in cell morphology were most evident in the administration of akalifa extract concentration of 100 microgram per milliliter. These changes in the form of shrinking cell size, cells appear to float, and the bonding between cells. Inhibition of cell proliferation and changes in cell morphology indicate cell death by apoptosis and necrosis. Cell cycle inhibition and apoptosis induction tests were carried out on the most active fraction that showed the highest cytotoxic activity, namely the hexane fraction of the akalifa. Cell cycle analysis was performed using the flow cytometry method to show the percentage of living cells in each phase of the cell cycle. The results of observing the cell cycle profile can be seen in Figure 2. From Figure 2, it is known that the treatment of the hexane fraction of the concentration of 40 and 80 microgram per milliliter does not affect the overall cell cycle profile of 41. Observation of the cell cycle with PI reagents to observe the effect of the hexane fraction treatment on the percentage of living cells in each phase of the cell cycle showed no significant changes compared to untreated cells, both in the sub-G1, G0 or G1S and G2M phases. Inhibition of the cell cycle in certain phases causes cell resistance to a chemotherapy agent, so the apoptotic induction mechanism is needed to overcome this. The treatment of hexane fraction concentration 40 and 80 microgram per milliliter in 41 cells showed the occurrence of late apoptosis, but the percentage was much smaller compared to untreated cells, as shown in Figure 3. This is presumably because the mechanism of the hexane fraction in killing cancer cells is without through the apoptotic pathway, but through the mechanism of necrosis. However, this conjecture needs to be further proven by trying the hexane akalifa fraction treatment with reduced concentration and longer incubation time. From the results of the research, we can conclude that akalifa ethanolic extract performed the strongest cytotoxic effect on 41 cells with the IC50 values of 171 microgram per milliliter. Among three fractions, the hexane fraction of akalifa showed the highest cytotoxic activity with the IC50 of 85 microgram per milliliter. Still, the fraction has no effects on cell cycle and apoptosis. This is the end of my presentation. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.